The overall goal of this procedure is to deposit a thin collagen assembly onto cells in a microfluidic device. This is accomplished by first modifying acidified native collagen through methylation to create net positively charged collagen molecules. The second step is to modify acidified native collagen through succinylation to create net negatively charged collagen molecules. Next, the microfluidic devices are prepared and seeded with hepatocytes, which are allowed to attach and spread overnight. The final step is to deposit the collagen assembly by alternately exposing the cells to the positively and negatively charged collagen solutions to create 10 bilayers of collagen on top of the cell layer. Ultimately, phase and immunofluorescence microscopy and albumin and urea assays are used to show the development and maintenance of cell polarity and secretory function.
The main advantages of this technique are that it enables hepatocyte culture in microdevices using similar techniques to those applied in plate cultures for over 20 years. It doesn't require complex device designs, and the deposited matrix is very thin, on the order of 100 nanometers.
We first had the idea for this method while brainstorming about how to translate classic hepatocyte cell culture techniques from plates to microfluidic devices. The collagen sandwich method works in open-top macroscopic systems, but the hydrogels used are incompatible with closed microdevices with limited channel heights.
Dilute 100 milligrams of a native acidified collagen solution to a concentration of 0.5 milligrams per milliliter with ice-cold sterile water, and place the solution on ice to prevent gelation. Next, adjust the pH of the collagen solution.